Good day. This is Fer and B. Pabale, the discussant of the topic Transcription and RNA Processing. The following are the definition of terms used in the topic that will be discussed, and is defined according to how it was used in the discussion. First is the protein synthesis. This is the building and making of proteins. Transcription is one of its processes. Messenger RNA or mRNA, the product of transcription process. Template strand, a DNA strand that RNA polymerase uses to synthesize mRNA. RNA polymerase, an enzyme that codes or synthesizes for mRNA from DNA strand. Promoter, where RNA polymerase begins or starts transcription. Elongation, is the stage when the RNA strand gets longer. Polyadenylation signal, a sequence of nucleotides that marks where an RNA transcript should stop. Termination, the process of ending transcription. Signal stop in RNA polymerase. 5' cap is a modified guanine nucleotide added to the first nucleotide in the transcript during transcription. Poly A tail, a 100 to 200 adenine nucleotides to the end of pre-mRNA. Spliceosome, an enzyme that removes introns and pastes together exons. Introns, junction sequences in the RNA molecule. Exons, good part sequences of RNA molecule that are assembled to make final and mature mRNA. Alternative splicing, a process that allows more than one mRNA can be made from the same gene. Transcription is one of the processes of protein synthesis. It is how we get the directions about how to make proteins from DNA and bring them to the ribosome so that ribosome can join amino acids in a specific order to synthesize proteins. So proteins are what matters. Our entire phenotype is based on the presence and absence of particular proteins. So simply, protein synthesis is how we build proteins. The process of protein synthesis called transcription is where DNA will be transcribed or copied into mRNA. In translation, the mRNA will find a ribosome and joined by amino acids and eventually form into actual proteins. But in this discussion, we'll focus in the process of transcription. So transcription happens in both prokaryotic and eukaryotic cells. Prokaryotic cells are in bacterium. Both transcription and translation happens in the cytoplasm because prokaryotic cells don't have the nucleus. And in eukaryotic cells, transcription first happens inside the nucleus, and then this mRNA leaves the nucleus, then the translation process followed. And in eukaryotes, mRNA will undergo a step called RNA processing, which is our second topic. And in this discussion, we'll focus on the transcription happened in the eukaryotic cells. In cells, we have our chromosomes. And in our chromosome, we have our DNA that contains our gene that are subject of the processes of protein synthesis. And take note that X-shaped chromosomes only exist after S phase of the cell cycle until the M phase when they separate. So I'll cover the other side of chromosome to avoid misconception. And only 1% of our DNA is our actual gene. A lot of our DNA is non-coding sequences, which means they don't code for protein. And this gene codes for our protein. So this is transcription. It is a synthesis of single-stranded RNA from double-stranded DNA template. It produces messenger RNA or mRNA specifically. And basically, this is the making of messenger RNA or mRNA. In transcription, it is where we copy DNA into mRNA. Transcription is an essential step in using the information from genes in our DNA to make proteins. Proteins are the K molecules that give cell structure and keep them running. Transcription uses one of the two exposed DNA strands. The one that it uses is called the template strand. This template strand serves as a template for synthesis of complementary RNA transcript. When we say complementary, is that they mutually supplying each other's slack. And also RNA's transcript is complementary to the template strand. And this RNA transcript is almost identical to the other DNA strand, which is the coding or non-template strand. Except that in newly made RNA, all of the T nucleotides or thymine nucleotides are replaced with U or uracil nucleotides. So this is an RNA or ribonucleic acid. It is a single strand, has a 5-carbon sugar called ribose instead of the oxyribose when compared to DNA. It has 4 nitrogen bases, the adenine, uracil, cytosine, and guanine. And compared to the DNA, it has uracil instead of thymine. One of the important characters in the process of transcription is RNA polymerase. The enzyme RNA polymerase synthesizes messenger RNA by trading one strand of DNA and follows base pairing rules to build the mRNA. RNA polymerase has multiple subunits. Humans and other eukaryotes have 3 different kinds of RNA polymerase, the 1, 2, and 3 polymerase. Each one specializes in transcribing certain classes of genes. For instance, the work of RNA polymerase, if there is a G in the DNA template, RNA polymerase will add a C to the new growing RNA strand. And RNA polymerase always builds a new RNA strand from 5' to 3' direction. So the directionality of polymerase. RNA polymerase synthesizes RNA strand in a 5' to 3' direction, while reading the template strand from 3' to 5' direction. The template DNA strand and RNA strand are anti-parallel. Always bear in mind that RNA polymerase reads template strand from 3' to 5', and that RNA is made from 5' to 3' direction. So how RNA polymerase know where to begin transcription? So transcription begins at initiation site. And in initiation site, there is this promoter region that comes before and slightly overlaps with the transcribe region whose transcription it specifies. It contains recognition sites for RNA polymerase or its helper proteins to bind to. The DNA opens up the promoter region so that RNA polymerase can begin transcription. And there is these promoters in eukaryotes. Helper proteins called basal general transcription factors bind the promoter first. And eukaryotic promoters have a sequence called a Tata box. The Tata box is recognized by one of the general transcription factors. It contains lots of A's or adenines and thymines, which make it easy to pull the strands of DNA apart. So after that RNA polymerase is in position of the promoter, the next step of transcription is what we call the elongation. Elongation is the stage when the RNA strands gets longer. During elongation, RNA polymerase walks along one strand of DNA, the template strand from 3' to 5' direction. So RNA polymerase synthesizes an RNA transcript, complementary to the DNA template strand in a 5' to 3' direction. It moves forward along the template strand in the 3' to 5' direction, opening the DNA double helix that it goes. The synthesized RNA only remains bound to the template strand for a short while, then exits the polymerase as a dangling string, allowing the DNA to close back up and form a double helix. RNA polymerase will keep transcribing until it gets the signals to stop. The process of ending transcription is called termination. It happens once the polymerase transcribes a sequence of DNA known as a terminator. Termination begins when a polyadenylation signal appears in the RNA transcript. Polyadenylation signal is a sequence of nucleotides that marks where an RNA transcript should stop. The polyadenylation signal is recognized by an enzyme that cuts the RNA transcript nearby, releasing it from RNA polymerase. RNA polymerase continues transcribing after the transcript is released, making 500 to 2,000 more nucleotides of RNA. Then detaches from DNA through mechanisms that are still under study. The molecule that directly made by transcription is called pre-mRNA, reflecting that it needs to go through a few more steps to become an actual messenger RNA. So it undergoes the process, RNA processing. So in RNA processing, there is an addition of 5' cup to the beginning of the RNA, and an addition of poly-A tail of A nucleotides to the end of RNA. And there is this chopping out of entrance or junk sequences and pasting together of remaining good sequences are called the exons. So both ends of pre-mRNA are modified by the addition of chemical groups. The group at the beginning is called the cap, while the group at the end is called a tail. Both the cap and tail protects the transcript and help it get exported from the nucleus and translated on the ribosomes found in cytosol. The 5' cap is added to the first nucleotide in the transcript during transcription. The cap is a modified guanine nucleotide, and it protects the transcript from being broken down. It helps the ribosome attach to the mRNA and starts reading to make a protein. So how is the poly-A tail added? The 3' end of RNA forms in a kind of bizarre way. When a sequence called a polyadenylation signal shows up in the RNA molecule during transcription, an enzyme chops the RNA in two at that site. Another enzyme adds about 100 to 200 adenine nucleotides to the cut end, forming a poly-A tail. The tail makes the transcript more stable and helps it get exported from the nucleus to the cytosol. Another RNA processing event that happens in the cell is what we call the RNA splicing. So in RNA splicing, we have our entrance. These are the specific parts of pre-mRNA, the junk sequences that must be cut out, and exons are the good parts version, the pieces of RNA that are not chopped out and pasted together to have final and mature mRNA that is shipped out of the nucleus. And splicing needs to be precise and consistent. This careful cutting and pasting is performed by the spliceosome, an enzyme complex made of protein and small RNAs. Most entrants contain marker sequences at both of their ends, which are recognized by small RNAs, and direct the spliceosome to remove the entron. Once the entron has been cut out, the spliceosome will glue or ligate the flunking exons together. A key point here is that it's only the exons of a gene that encode a protein. Not only do the entrons not carry information to build a protein, they actually have to be removed in order for the mRNA to encode a protein with the right sequence. If the spliceosome fails to remove an entron, an mRNA with extra junk in it will be made, and a wrong protein will get produced during translation. Splicing seems like a wasteful system, and that the reason of splicing is still under study. However, splicing does allow for a process called alternative splicing, in which more than one mRNA can be made from the same gene. Through alternative splicing, humans and other eukaryotes can sneakily encode more different proteins than the genes in the DNA. In alternative splicing, one pre-mRNA may be spliced in either of the two, or sometimes many more than two, different ways. For example, in the diagram below, the same pre-mRNA can be spliced in 3 different ways, depending on which exons are kept. This results in 3 different mature mRNAs. each of which translates into a protein with a different structure. A sequence of DNA encodes a pre-mRNA transcript that contains 5 regions that may potentially be used as exons. The exon 1, exon 2, exon 3, exon 4, and exon 5. The exons are arranged in linear order along the pre-mRNA and have entrons in between them. In splicing event number 1, all 5 exons are retained in a mature mRNA. It consists of exon 1, 2, 3, 4, and 5. When it is translated, it specifies protein A. In splicing event number 2, exon 3 is not included in the mature mRNA. It consists of exon 1, 2, 4, and 5. When it is translated, it specifies protein B. In splicing event number 3, exon 4 is not included in the mature mRNA. It only consists of exon 1, 2, 3, and 5. When it is translated, it specifies protein C. So here is my references, and that would be all. Thank you.